Hello, college biology students. I'm going to go through some of the functions of the membrane proteins today. We're going to talk about the functions of those membrane proteins, explain how proteins can associate with the membrane itself, and then identify some or a transmembrane domain. We kind of hinted at this earlier. Cell membrane proteins, and you can see I have 6 functions listed here. And this visual is of enzymes, which we're going to talk about. So again, mentioned earlier, some of those membrane and bound proteins. So like that's what these blue things kind of represent, are all the proteins embedded in the membrane, and you can see their different shapes, and they have different functions. So some of them are just transporters. Remember, we said that cell membrane is selectively permeable. It only lets certain things through. So sometimes certain membranes are only going to recognize a very specific molecule that it allows to go through. So that's one of the functions. Also enzymes. Remember, we said enzymes are catalysts. So enzymes are catalysts that speed up chemical reactions, but they're biological catalysts. Sometimes in chemistry we talk about chemical catalysts, but these are actually biological catalysts. And this, just a quick review. So catalysts are protein molecules that have a very specific shape. So on here, the dark color represents the enzyme. The yellow golden kind of color represents a substrate. And so this process is very much like a lock and key. So, and a very specific substrate will fit this particular enzyme. It attaches. As soon as that bond is broken, it breaks it into 2 smaller substrates. Then it releases it, and this enzyme goes and just looks for another one of these substrates, and it gets used over and over and over again. Okay, so there's a lot of enzymes actually in the cell membrane. Some of those proteins are actually embedded in the cell membrane are actually enzymes. Cell surface receptors. We talked about cell signaling earlier. I think the end of chapter 4. So the cell surface receptors are there to detect certain chemical messages, and sometimes we talked about how hormones can be important in sending. Are one of the things that are important in used to send messages generated from cells in one part of the body, all the way to cells in a different part of the body. So these are usually our long range chemical messages. There are some other ones that are short range messengers, and of course there's electrochemical signals that go between nerve cells as well. Cell surface identity markers identify cells to other cells. And some of that, remember, we said, is recognizing self and non-self. Sort of like, do you belong in this body or not? Or are you a foreign particle? So some of those markers are to identify that. Like, nope, I'm really one of you guys. Like, I got the right markers, so don't attack me. We also have there's tons of different kinds of identity markers. There's different receptors and so forth. We talked about this in terms, just briefly, I mentioned this in terms of determining blood type. But there's also, with certain cancer treatments, they can identify whether your, say, B cells, which are part of your immune cells, have certain receptors, and they can use certain medications or chemotherapies that target certain cell markers. So like if you have B cell lymphoma and you happen to have this CD20 cell marker on your B cells, and the cancer is in your B cells, then they can target that CD20 marker with different medications or chemotherapy agents. So, so sometimes we use that in terms of treatment as well. Cell-to-cell adhesion proteins, we talked about how a lot of cells are, you know, if you look at tissues in that whole matrix, those cells are all connected to each other. So some of those proteins are just simply to connect to other proteins on adjacent cells to kind of hold that mass of cells together. And then we, and I know we mentioned this when we talked about cell communication, attachment to the cytoskeleton. So some of those cells, remember the cytoskeleton? Kind of those pillars that provide support. Maybe some of those attached, you know, they attach to proteins, which then attach to cytoskeletons on the next cell and so forth. So these anchor proteins, we call them, are used for attachment. And then some other ways that things move through the cell membrane. We're going to talk about some types of diffusion, the different types of channel proteins, and then, of course, osmosis, which I kind of had you do that dialysis tubing lab a little bit ahead of time before we really talked about this, because as I told you, I felt like every day in the classroom was kind of a gift, and I didn't want to waste that with lecture time. So I'm wasting your time now with lecture time, but you do need to know this. So diffusion, and if you'll remember, like if you had me for 10th grade biology, you've seen my little diffusion. I don't know if you can see, I dropped some food coloring in here. Oh, I'm getting a lot of reflection. Some food coloring in here, which...
Kira Wolpert to the front office, please. Kira Wolpert to the front office.
Sorry about that. So I have my food coloring in here, and even though you can actually see this is really turning pretty colored already, let's go to white. So now you can see a little bit more the my diffusion. So I had just plain water that I put some food coloring in, and I really tried to hold it as still as possible. And regardless of, sorry, this mirror image here, regardless of that, we can really see this food coloring kind of move through here just due to the random molecular motions that occur, even if I'm not moving this around. So that would be just simple diffusion. Well, some of that happens in our cells as well. So we definitely have things moving through the cell membrane that occur just simply because of diffusion. So when I dropped my food coloring into my beaker of water, at first that food coloring was all coalesced into one little drop or a couple little drops all together. But over time, due to random molecular motion, it began to spread and spread and spread out. And it's going to continue to spread out until it's equally dispersed throughout the beaker. And that doesn't really take energy. It's just due to the random motion of the molecules that are in the water. The food coloring is definitely going from an area where it's more concentrated to an area where it's less concentrated. So it's going from that coalesced first drop and spreading to areas where there was no food coloring. And it's just going to keep doing that because nature doesn't like that concentration gradient. So we have this happening in our cells as well. And it's going to keep doing that until we say equilibrium is achieved and there's equal concentrations throughout my beaker. And things like carbon dioxide and oxygen will move that way through cell membranes until there's equal concentrations of oxygen and carbon dioxide on both sides of the membrane. And we'll talk about that more when we learn about respiration. Hi, sorry, I had to move from my room and now my camera won't come back on. Don't look at me. I'm a mess. Anyway, I'm sitting in my back office, and a bunch of 8th graders are in our room. So anyway, so carrier proteins then are molecules that are proteins that attach to certain molecules and carry them through the membrane. So like if this is a carrier molecule right here, we outline this in yellow. I'm drawing with my mouse now. So if this is the carrier protein, you'll see that it's a peripheral protein. It doesn't extend all the way through the membrane. So it will attach to some sort of cool little molecule. So whatever this cool little thing is here, it's going to attach to only little pink square molecules or whatever the particular molecule is. It's usually very molecule specific. That's going to attach, and then it's going to drag this through to the other side of the membrane, whether that's into or out of the cell. And then channel proteins in the cell membrane that span the entire membrane, creating a channel that usually kind of opens up or is open, more like a tunnel that allows things to move through. So this, again, from our picture before, this would be an example of that channel protein you can see, and sometimes they're pretty specific, and sometimes they will transport maybe positive ions of a certain size or something like that. And then moving on, facilitated diffusion. So when we did our, I just want to go back really quickly. So when we did our dialysis tubing lab, we were just really looking at simple diffusion through our little cell made of dialysis tubing. So things that were molecules that were large, like starch, really couldn't pass through this membrane, but other things could move through this membrane. This isn't a super accurate representation because, and I don't remember if I mentioned this, but something like glucose really can't pass through our cell membrane, but it has to get into our cells. So it has a carrier molecule, and that's actually what insulin is. So if you are lacking insulin, you need to take injections of insulin. So if you know anyone who's diabetic, that's actually the case with them. Facilitated diffusion is passive transport. So active versus passive transport. Active transport requires energy. Passive transport does not. So simple diffusion is really a type of passive transport. So I guess just really quickly, so when we talk about passive transport, and if you think of someone who's passive, they're usually pretty low key. They don't get riled up about things. They don't get super energetic about things. Where someone who's very active uses a lot of energy, and that's the same with movement through a cell membrane. So passive is like the diffusion when I put food coloring in water, like that's obviously very, very passive. So facilitated diffusion is a type of passive transport in which carrier proteins are used to transport certain substances through the cell membrane, but it's still moving from an area where there's more of the substance to an area where there's none or less of the substance, just like my food coloring in my water. We also would say ion channels. Again, this is a type of passive transport in which, things are still moving, with that concentration gradient. Sodium potassium pump is one example of that. You have to really regulate the sodium and potassium in your cells. We do that with the sodium potassium pump. And I think I'm not going to have enough time to get through all of osmosis today, but just know that when we looked at our dialysis tubing, on that third day, because remember we set it up on a Wednesday, looked at it on Friday. On that Friday, most of you or a lot of you noticed that, wow, they're actually the little cell made membrane made out of dialysis tubing seemed to look a little fuller than when you originally sat it up. And that was because water moved into the cell. So that tells us that the inside of the cell was a hypertonic environment and was higher in solutes than what was in the beaker outside of our little makeshift cell. So we'll talk about this tomorrow, or rather on Monday. Have a great weekend.